This video covers notes for DNA structure and replication. We'll start with describing what DNA is. DNA stands for deoxyribonucleic acid, DNA. DNA is a nucleic acid, which, remember, is one of our 4 categories of organic molecules. And this means it is made of nucleotides. The function of DNA is that it holds the code for the characteristics expressed by an organism. So it has the instructions. The shape of DNA is a double helix. So it's like a twisted ladder, sort of a thing like this. And those 2 strands are held together by hydrogen bonds, as we'll see later. The location of DNA is different depending on the cell type. So in eukaryotic cells, DNA is found in the nucleus, and it is linear. So it's a bunch of strands, but they're in linear segments. In prokaryotic cells, DNA is found in the cytoplasm, and it is circular. So both ends of the DNA are connected there. Let's look next at the structure of DNA. As we said, DNA is made up of nucleotides. Each nucleotide contains a phosphate group, a sugar, which in this case is called deoxyribose, and a nitrogen base. So what we see here is the basic structure of a nucleotide. We have a phosphate that is connected to deoxyribose sugar, which on the other side is connected to a nitrogen base. And these nitrogen bases can be one of 4 different things. In DNA, there are 4 different options for the nitrogen base. A for adenine, T for thymine, G for guanine, and C for cytosine. Those are the 4 nitrogen bases. So what's left on that end could be one of those 4 things. And remember that DNA is double-stranded, so there's going to be 2 nitrogen bases across from each other on either side of that ladder shape. And those are going to be held together by hydrogen bonds. What we know about DNA is that A and T match up and are held together by 2 hydrogen bonds. And G and C are held together by 3 hydrogen bonds. So I'm going to draw over here to the side kind of a untwisted ladder type shape. So if these are the outsides, that's my sugar and phosphate. On the inside, we'll have A and T across from each other that are held together by 2 hydrogen bonds. That's what that dot is. And then G and C are held together by 3 hydrogen bonds. So again, if we untwisted that double helix, that's what we would see in a very basic sort of way. Nitrogen bases can be two different shapes. A and G are called purines. T and C are pyrimidines. And so what you'll notice by how the bases match up is that a purine is bonded to a pyrimidine every time. A purine is a 6-sided thing that's attached to a 5-sided thing. So it's two things. And then pyrimidines just have the one. And so one of the really cool things about DNA is that the distance from here to here, this distance is going to be equal all the way up and down because every time we have a purine connected to a pyrimidine. I know that's just extra info, but I think it is one of the super cool features of DNA. Next, we'll look at the specifics, wrong way, the specifics of deoxyribose. So I need you to bear with me because I'm about to say a lot of words, but I think you're going to be okay. All right. Deoxyribose is a 5-carbon sugar. So it has 5 different carbons, and each one of them is numbered. And based on every single time, it's numbered in the same sort of clockwise way. And we know based on which number carbon it is, what is going to attach to what. So here we have carbon number 1, number 2, number 3, and number 5. Our big focus there's 4. Our big focus is going to be on the 3rd and 5th carbons. Those are really important attachment points for how DNA attaches itself together. So the phosphates attach to the 5 prime, we call them prime, the 5 prime carbon over here. So attached to this carbon is where the P for phosphate is going to be. And then the 5 prime end of one nucleotide is going to bond to the 3 prime carbon of the next nucleotide. And this creates a sugar phosphate backbone. Over to the side, I'm going to show you what I mean. Please admire my shape drawing. I'm going to start just by drawing 3 sugars and then show you the attachment points. Right. So here is my sugar. I have my phosphates over here, and then my nitrogen bases over here. And then we'll have the, flip-flop reverse of this on the other side, but I'm going to have the attachments like this from 3 prime end to 5 prime end. DNA strands together are called anti-parallel because they are, again, the flip-flop. So one strand is going to be 5 prime to 3 prime. And again, we're talking about the orientation of that sugar. And the other one is going to be 3 prime to 5 prime. So one of them looks kind of upside down, which we'll see here in just a second. And that is why they're called anti-parallel, because they are flipped. So let's look at this diagram of DNA. So I can see that these purpley ones are thymine, green is for adenine, bluish is for guanine, and red is for cytosine. This segment right here is the phosphate. And this portion right here is the sugar. So on this side, this strand is running 3 prime to 5 prime. And the one over here is 5 prime to 3 prime. Again, that's just the orientation of the sugar. So there we go. moving on to how did we ever figure out what binds to what in DNA? So, although Watson and Crick get the credit for the discovery of the structure of DNA, a lot of scientists' work went into it. And one of them was that of Erwin Chargaff. And he developed a rule that says, if we are analyzing the percentages of A, G, C, and T in a DNA sample, what he noticed was that the percentage of adenine and thymine are the same, and then C and G are also the same. So that led him to the conclusion that A must bind or pair with T, and C pairs with G. So his rule says that DNA samples will have the same percent of A and T, and also the same percent of C and G. So let's look at an example problem here. So let's say that we have a sample of DNA that is 32% adenine. What I know is that that same sample, so let me make a note, we have 32% A. That also means I have 32% of thymine as well. So that's one thing, check that off. And so then how do I figure out how much C and G are left? So I know I have to have 100%, and I've accounted for 32% and then another 32%, which leaves me with 36% unaccounted for. So I know my DNA sample is 30%, but that's C and G. So then I do 2 into 36 to get 18. And then I know my sample is 18% C and 18% G. That is all we need to know for now about the structure and function of DNA. What we need to move on to next is DNA replication, which describes how DNA is able to make a copy of itself. Because when cells are getting ready to divide, we want to make sure every single cell has all of the same DNA in it. We don't want to have less or only half or something. We want to, maybe as your body is making more skin cells, for example, every skin cell needs to have the same DNA. And so DNA replication is the process by which DNA makes an identical, that's an important word, an identical copy of itself. So we are making exact copies of DNA. And again, the location is going to be different in eukaryotic and prokaryotic cells because this process is going to happen wherever the DNA is housed. So in eukaryotic cells, replication occurs in the nucleus. And in prokaryotic cells, it occurs in the cytoplasm. And that's just because that's where the DNA happens to be. The purpose of DNA replication is to make sure every cell has a complete set of DNA. This process is going to happen before both mitosis, which is coming soon, and meiosis, which is coming soon, but not as soon as mitosis. Before we get into the details of replication, I want you to be familiar with 3 important enzymes that are essential for DNA replication. The first one is called helicase. Helicase unwinds / unzips the double helix. So our double helix is this like squiggly shape here. We want to open that up before replication can occur. So helicase is going to break those hydrogen bonds and open up that double helix. The next enzyme that is important is called DNA polymerase. And the job of DNA polymerase is to add new nucleotides. So what, as a preview, is going to happen is once our helix is open, DNA polymerase is going to read one side and see, oh, there's an adenine here, better put a thymine on this new strand that I'm building. And then finally, ligase connects fragments of DNA. Because there will be some. So let's go through the steps of DNA replication. So the first thing is that helicase is going to unwind and unzip part, not the whole thing, just part of the double helix to form the replication fork. So below we have a pretty simplified version of DNA that literally looks like a ladder, but we do have our 3' and 5' ends labeled. Remember, that's just which way the sugar is flipped, because that will be important in DNA replication, because replication is not going to happen in the same way on both strands. So what's happened is, here's our helicase. It is moving along the DNA molecule and unwinding and unzipping that. And this open space right here is the replication fork. Right there. So that is going to happen first. Next, scroll down here. DNA polymerase adds nucleotides to both strands. So essentially what's happening is we're going to open up the thing and then replicate on each fragment, or on each strand. So we're using both strands as a template to create a new strand on both sides. And that's going to happen differently depending on the strand that it is. So if we look at our diagram down here, the strand that is 3' to 5' is called the leading strand. And the one that is 5' to 3' is called the lagging strand. These names are a little bit misleading because replication, if we look at the speed, is about the same on both. This lagging strand is just a little bit more complicated. And the issue here is that when new nucleotides are being added, they can only be added from the 5' and towards the 3' end because of how the sugars attach. They can only be attached to that free 3' end. So on the leading strand, replication occurs from 5' to 3' continuously, which we can see in the diagram here. It's just able to add those nucleotides because the sugar is in the right direction. On the lagging strand, replication occurs in segments. It requires a primer to get started. which you can see here, that's this little red piece. Those are the primers that say, all right, I'm going to attach right here, and then you can replicate. And then each of these segments is called an Okazaki fragment, named after the scientist that kind of discovered this. And then finally, DNA ligase connects the fragments. So eventually, this piece down here is going to be one continuous piece, but only after ligase is going to make a connection here. So the leading strand, we can add from 5' to 3' continuously, no problem. On the lagging strand, it has to be in fragments because replication has to occur in the 5' towards the 3' end. The last thing is that DNA replication is called semi-conservative. Because each copy of DNA will have one old strand and one new strand. So let me show you what I mean by that. So let's say this is in blue. This is my original DNA molecule. After this molecule has been replicated, the two new ones will be part old. So there's my outside pieces of that. And then they will also be part new, where the pink here is my new strand. So it's semi-conservative because it's only conserving half of the old one. I guess let me put a plus sign. We're using the old strands or the original strands as a template to create two new ones. But, so even though we contain one old and one new, these two things are still identical. And that is a not so short summary of DNA structure and replication.